Hi, I'm Anthony Barley, and I'm a member of the 2015 Arnold O. Beckman Postdoctoral Fellow class. My goal in this video is to give you a brief introduction to the research that was enabled through this generous gift through the Arnold and Mabel Beckman Foundation.
I've always been fascinated by the diverse types of organisms that coexist with us on Earth. Species are the basic unit we use to quantify and measure biodiversity, and thus an understanding of the boundaries between species is a fundamental piece of information for all fields across the life sciences, from ecology, conservation, and development to agriculture, pest management, and medicine. One of the simplest, but also most challenging questions scientists have struggled to answer over the past couple hundred years is, how many species of organisms exist with us on Earth? In 1758, Carl Linnaeus, who formally developed the foundations of modern biodiversity science, hypothesized that there were probably about 12,000 species on Earth. Speculation continued until the 1970s, but up to that point, scientists doubted that there were many more species on Earth than had been formally cataloged and described, which at the time was about 1.5 million. Then, in the 1980s, scientists began to wonder if they could bring some real data to bear on this question. In a particularly influential study, an entomologist at the Smithsonian Institution exhaustively sampled all the species of beetles that existed on 19 trees in the Brazilian rainforest. Based on this data, he estimated that there were probably 30 million species of tropical arthropods on Earth alone, suggesting that the total number of species on Earth is closer to 100 million. Since then, scientists have continued to come up with creative approaches to address this question more accurately. Although the debate remains unsettled, one thing is clear. There are many more species that exist on Earth than are currently known to science. As species extinction rates climb, due to our growing human population, addressing this question has become more important than ever. The genetic revolution of the past several decades has served to highlight this shortfall in our basic knowledge of global biodiversity, because new DNA sequence data has revealed many new species and branches in the tree of life that were previously unknown to science. However, genetics has also been suggested as a means for addressing this knowledge gap. Given the ease with which we can now sequence the DNA of any organism, if we can accurately identify the boundaries between species based on genomic patterns, this would provide an efficient means to solve this fundamental question about Earth's biodiversity and ultimately address a major goal for the life sciences in the 21st century. Around the time I was finishing graduate school, a number of scientists had begun developing approaches to identify the boundaries between species using genetic data. The key to this lies in the development of statistical models that describe the way in which genetic changes occur during the process of species formation. Although a number of different approaches had been proposed and were in wide use, no one had developed a formal framework for testing how well these models actually describe the processes that shape real genetic datasets from natural systems. It is widely known that if statistical models do not sufficiently describe the processes that have produced observed data, in this case, DNA sequences, inferences drawn under those models are subject to bias and inaccuracy. Being interested in this question, I wrote a postdoctoral fellowship to the Foundation, asking that they support my quest to develop new methods for assessing the fit of these models to real empirical datasets. The first two questions to be addressed were, do we come to different conclusions about species boundaries when we use different statistical models? And if so, does this result reflect differences in how well the models describe the biological processes that occur during the process of speciation and shape genomic patterns? Through assembling a large series of exemplar genetic datasets from many different groups of species on Earth, I demonstrated that indeed, the use of different models fundamentally changes the biological conclusions you come to about species boundaries. By simulating datasets under a wide range of biological scenarios thought to occur in nature, I was also able to show that statistical models vary in their sensitivity to violations of the assumptions they make about the biological processes that have occurred. Finally, using a general statistical framework known as posterior predictive simulation, I developed new methods that can be used to assess the fit of different models to empirical datasets, so that researchers can determine if they should be concerned about the suitability of a particular model for drawing inferences in their individual datasets. These new computational tools employ a common framework. First, we perform inference under our model to obtain estimates of the model parameters, including in this case, the number of species in a dataset. Next, we use those parameter estimates to fit our model to a dataset and simulate new predictive datasets that can be compared to our original data. Using test statistics that quantify important aspects of the data, we can assess if the predictive datasets are similar to our original data, in which case our model effectively captures the biological processes that have shaped our data, and we can be more confident in our inferences, or if they are very different, in which case our model is not likely to be a good match to our data, and we should be concerned that our inferences may be biased. I published this research in a couple of the top journals in the field of ecology and evolutionary biology. My results also highlighted that one of the major challenges to the performance of these models are violations of model assumptions about gene flow between species. The genomic revolution has highlighted the fact that many species exhibit limited amounts of genetic exchange due to low levels of hybridization between them. In the final year of my fellowship, I began developing a group of lizards in which hybridization is remarkably common as a model system for understanding the impact of gene flow on genomic patterns. The goal of this work is to use the system to inform the development of approaches for identifying species in these types of scenarios and better understand how hybridization shapes the evolutionary history of species in nature. Using preliminary data I collected as a postdoctoral fellow, I co-wrote a grant to the National Science Foundation to continue this research, which was recently funded.
Thus, I want to extend my deepest gratitude to the Arnold and Mabel Beckman Foundation for supporting my research. I hope that I have effectively demonstrated how their support has contributed to the development of important new tools and knowledge related to addressing important questions in biology, as well as help launch my career as a young independent scientist, both of which I believe are consistent with the goals of Arnold Beckman and the legacy he hoped to achieve in starting the foundation.